Hello friends, with the continuation of previous discussion regarding the significance of meiosis, how far the meiosis is different from mitosis, and today's discussion we will continue in knowing what exactly meiosis is and what are the events that are going to happen in meiosis. As I said, the meiosis is going to have two subphases. One is going to be the meiosis 1, other is going to be the meiosis 2. So in today's discussion, we will just have a discussion of the events that are happening in meiosis 1. As I said, the major difference that exists between the meiosis and mitosis is going to be with meiosis 1, and that too the prophase 1. So let's get into the discussion what exactly is going to happen in meiosis 1, right? So as I have said, the meiosis is going to get divided into two phases, that is meiosis 1 and meiosis 2, right? So this meiosis 1 is further divided into prophase 1, metaphase 1, anaphase 1 and telophase 1, right? And you know that at the end of meiosis, the ploidy, the parent cell, which is going to be 2n, is getting reduced to haploid. Hence we call meiosis as a reductional division, right? Reductional division. So all these concepts were elaborately discussed in the previous video. So kindly get back to that video in case if you have not viewed it, right? Fine. Now among these, one of the important subphase here is going to be with respect to the prophase 1. This prophase 1 is going to have further subphases. Say this prophase 1 is going to have leptotene phase, zygotene phase, packetine phase, diplotene and diakinesis. So these are all going to be the subphases in under the prophase 1, right? So though these terms might create a sort of apprehension, but friends, the concepts are going to be as simple, okay, as we think, right? So once we understand the things that are happening here, getting into these subphases conceptually is not a big task here, right? So let's begin with the leptotene stage. So the picture is being given here. This is going to be the overall picture, and this is what is going to happen in prophase 1. So let's dive deep into what exactly happens in prophase 1. Then after that we will come to this overall picture, right? Okay, say. So leptotene. So what happens in mitosis, prophase of mitosis? If I say just prophase, it means that the mitotic prophase. So in case of meiosis, we will call it as a prophase 1 and prophase 2 with respect to meiosis 2. So if we simply say prophase, then it is a mitotic prophase. So what exactly happens in the prophase of mitosis? You know that the chromosome condensation begins, centriole moves to the opposite pole, it is going to radiate the mitotic spindle, so all those. So everything what you have discussed in that prophase is going to happen in this prophase 1, but with some extra events which is getting dispersed into these subphases, right? The exact thing that is going to happen in prophase is also going to happen in prophase 1 with some additional events, and all these are going to get dispersed into various subphases like leptotene, zygotene, packetine, diplotene and diakinesis, right? See, in that what is going to happen in leptotene phase? In leptotene phase, the chromosome chromosome condensation begins. right? This is one important event that is going to happen in leptoteen. The chromosome condensation begins. The chromosome becomes a visibly compact structure when viewed under the microscope, right? This is going to happen in leptoteen stage, which is going to be the beginning of prophase 1 of meiosis 1, right? And maybe the centriole can move to the opposite poles, right? Start radiating the mitotic spindle, the spindle fiber, So such initial events can be expected in leptoteen stage, right? Fine. Next, what is going to happen in this zygotine stage? See, zygote. Where you have heard the term zygote. What is zygote? It is nothing but the fertilized product of the male and female gamete. The male gamete and female gamete both get fused to form zygote. So zygote means union. So here also, zygote, it is easy to remember, it is a subphase where some union is going to happen. So what is that? The homologous chromosome, right? Homologous chromosome is going to combine. So if this is not visible, I will make it in the homologous chromosome is going to unite, combine. That is what is called as zygotine here. And you have to know, for this you have to know what is homologous chromosome. So I have made a separate video regarding the basic terminology. So kindly go back to those videos, get these concepts, get this term cleared, and then join us here, right? So let's say this is going to be the homologous chromosome, right? Homologous chromosome. This is in monad. This is in monad. I can call this as C1F, and this is going to be C1M. Since this has crossed the S phase, it has got the S phase, because before this meiosis 1, we would expect the interphase should have happened, right? So interphase is going to be the preparatory phase both for mitosis as well as meiosis, right? So in the interphase you have the synthetic phase. So during synthetic phase, what do you expect? The chromosome, the DNA duplication happened, the chromosome concentration is increased, but the chromosome number remains the same. So what happens? I expect one more chromosome getting added here, right? So the chromosome concentration will increase, but the chromosome number will remain same, right? Chromosome number will remain same. So let's keep like this. So this is going to be dyad, right? This is going to be dyad, right? So I call this as a C1F, and this is going to be C1M. So this homologous chromosome combination or the union is what is going to happen in zygotine stage, right? So wherever the homologous chromosomes are there, say C1F is getting paired with C1M, C2F, okay, chromosome 2 of father is getting paired with the chromosome 2 of the mother. So this union is what is going to happen, and this is enabled by a ladder shaped structure which is called as SC complex, and the SC can be expanded as synaptonemal complex, right? Synaptonemal complex, right? Okay. So this is what is going to happen in zygotine stage, right? So this is done. So you could easily remember the event in zygotine phase as the zygo means union. Here the union of two homologous chromosomes are expected here, right? And the next phase is going to be the Pachytene stage, right? One important event that is going to happen in Pachytene stage is is crossing over. Okay, one important event that is expected here is crossover. Say what happens here is, this is C1F. Let me call this as A chromatid, B chromatid, C chromatid, D chromatid. Now what happens? See friends, before you get here again, I expect you should know what basic difference that exists between the homologous chromosome. That is, these are going to be the non-sister chromatid. This and this are going to be the non-sister chromatid, right? Okay, maybe I can put this in a different color that should be instead of blue, let it make this color, which will be more easy for us to remember. Instead of this, I'm going to make it in a pink color, right? Okay, pink color. So these are going to be non-sister chromatids. Non-sister chromatids I have given the similarity as well as differences. Sister chromatids A and B, C and D. These are sister chromatids which are going to be same. Non-sister chromatids are going to be partially same. That is same with respect to the gene profile, but allele will be different. Say for example, it could be T here, here also it can be T with respect to height gene. This is going to be small, this can be small t, this can be small t. So the gene profile is same, but allele is going to be different. Friends, I am not here to explain these things because I have done a previous video already explaining the similarities and differences, right? Now what happens is crossing over. What is crossing over? A part of the chromosome will split, okay, here, okay, and this C might go to B and B will come to C. So the C will go to, B will go to C, and in turn the C will come to B. Right. So what profile initially we had? Capital T, small t, small t. Now after this crossing over, you see capital T, small t, okay, and capital T, small t. Right. Initially we had the allele capital T, small t, small t before the crossing over. Now after the crossing over, this small t, this chromatid has come here. That is C has come to B, B and B has gone to C. So you see capital T, small t, okay, and capital T, small t. So this is what after recombination, and this is going to be before recombination, right? That is the crossing over, okay, B going to C, C going to B, that is crossover. It is going to change the combination. What combination? Initially capital T, okay, now it has become capital T, small t. So this crossing over lead to recombination, and this is one important event that you are supposed to remember because it is going to play an important role in case of linkage that will be discussed in genetics, right? So this is going to be the recombination, right, crossing over, and you have an enzyme called as recombinase that is going to facilitate this, what is that, the crossing over or the recombination. Crossing over is the event, the output is going to be the recombination, right? Now hope you are clear with this. So if at all you might have some difficulty, it might be with respect to the terminology. So I repeat, please go through the previous discussion, get these terms familiar. In case of you have any doubt, kindly get it clarified so that you will be able to understand these concepts with utmost clarity, right? Fine. Now what is going to happen in diplotin? Diplotin, what happens? See, it is going to be somewhat reversal of zygotin. What is zygotin? That is going to be the pairing of homologous chromosome. Now what happens is the synaptonymal complex is going to dissolve. The SC complex is going to dissolve so that the paired homologous chromosome is going to separate apart. Okay, but they are not completely separate. Wherever the recombination happened, okay, still the chromosomes are associated or attached at that particular point by an X-shaped structure which is called as chiasmata, right, right? Hope this is clear. So in short, in diplotin stage, the homologous chromosome, paired homologous chromosome separate, but still by the dissolution of synaptonymal complex, but still they are associated at some ends wherever the recombination has happened by an X-shaped structure called as chiasmata. This is what is going to happen in diplotin, and diakinesis is going to be the terminal phase of prophase 1, where the X-shaped structure is going to is moved towards the end. Say for example, here is what the chiasmata is formed, right? So this is going to get the chiasmata X-shaped structure is going to get towards the end of the chromosome, which you call it as a process called as terminalization. Okay, you call this as a terminalization. So this is one step, right? And second thing is the nuclear membrane disappears, the nucleolus disappear. Okay, so and as you know that the disappearance of nuclear membrane is going to mark the end of the prophase, right? So this is a short summary right, of the prophase 1. So this is the major difference, friends, that exists between the prophase and prophase 1. Right. So hope you have understood these things. Now in short, at the end of prophase, the homologous chromosome, right, they are somewhat paired. They are not completely separate, they are not completely paired. Okay, pairing has happened and the repulsion also has happened, right? Fine. Now with this, let's get into the discussion. Now you could see that this picture is going to show you the pairing of homologous chromosome, right? The recombination, everything has happened. Now this is the prophase 1 which we have discussed. Now coming to metaphase. Now what happens in metaphase? The paired homologous chromosome, they are going to get aligned at the center of the equatorial plate, right? Equatorial plate. So this is, yeah, this is one chromosome. Let's call this as C1F. This is going to be C1M. Let's call this as a C2F. This is going to be C2M. So they are going to get aligned. This is what is called as a metaphase plate, as same as what we have seen in mitosis, and you could see the centriole radiating the spindle fiber, right? Now friends, one important difference that you are supposed to note here is, in case of mitosis, see for example, if this is going to be the chromosome which has got a centromere, both the end of the centromere is accessible for the spindle. Now since the pairing has happened, see this centriole, the spindle fiber from this centriole, let's say I can call this as a south, like this as a north. The north will have access only to the one end of the kinetochore, because the other kinetochore is not accessible to the spindle fiber. Hope you get this point, right? Okay, since my point is, since usually this is a kinetochore, assume that this is a kinetochore. This is a kinetochore which has got, this is a centromere which has got two ends, kinetochore. So one spindle can attach here, other spindle from the centriole can attach here. But since the pairing has happened, this kinetochore is masked. This kinetochore is masked, right? Only the other kinetochore is open, right? So obviously, what happens? Okay, the spindle from the one centriole has got an access only to the one kinetochore, and the other kinetochore is masked. Likewise, this spindle from this centriole has got access only to one kinetochore, right? Hope I am not confusing you, right? In short, there are two kinetochore. Due to pairing, one end is closed, the other end is open, so the spindle will attach. Whereas in mitosis, the spindle will have access to both the kinetochore since pairing has not happened, in simple terms, right? And what is going to happen in anaphase? Anaphase, the centriole is going to, the spindle is going to pull the chromosome, so it is going to move towards the pole. Okay, you could see that. Okay, that is here. You could see the the entire chromosome moves. Right, there is no splitting of chromosome here. Okay, this is another important difference. Whereas, say, if you see a chromosome in case of mitosis like this, what happens? One spindle is attached here, the other spindle is attached from the centriole, and there is going to be a split in the split in the chromosome. Whereas here there is no split happening, the entire chromosome is getting moved towards the pole, right? And at the end of telophase 1, a separate nuclear membrane, everything is formed, okay, within a cell. Now you could see that this is going to be C1F, this is going to be C2F, and this is going to be C1M, this is going to be C2F. So the ploidy is getting reduced to haploid. Now, if in case of cytokinesis is going to happen, this is going to be a haploid, one haploid cell, right? And this is going to be the another haploid cell. So, as we have said, the meiosis one is going to reduce the ploidy, and the meiosis 2 is going to further begin with these two cells, right? See friends, this is a bit complicated where you have to understand the basics for you to understand these events, right? So that's the reason why I have taken the previous video and explaining the basics of all these concepts, without which this is going to be difficult, right? So I hope I have tried my best in conveying these things. So please go through the video. In case if you have any doubt, get it clarified, right? So we will meet in another video continuing what is going to happen in meiosis II, right? See, the key point here is you have to, the concept is all going to be same. Here and there, there's going to be some differences, right, between mitosis, meiosis I and meiosis II. So once you understand all those differences, then you can apply the generalized concept and then understand these things with utmost clarity, right? So let's stop the video here, right? So in next video, we will try to proceed with meiosis II, right? Hope you have understood this. Join us in the next video. Thank you.